This lecture is going to focus on organic polymers, and that term might sound foreign to you, but you actually know what these are. These are things like carbohydrates and proteins, lipids, which are fats, and nucleic acids like DNA, RNA. Polymers are these gigantic compounds where you have molecules, these repeating subunits of molecules that go ahead and bind to one another. You can see it's a strong bond, it's a covalent bond. So these are long strings of molecules, and so they're called polymers. What monomers are the repeating subunits. You can see here's this one with the little green guy on it, and here it is right here, so that guy's right there. So these line up in a particular order to create a much, much larger compound. Now there's a couple reasons why this is set up this way. If you think about food that you eat in your diet, let's say you eat protein, okay? You have some protein that comes through, and it comes down the track, and you're going to have to do something with that protein, right? Ideally, you want to absorb all the amino acids, right? But this protein is a long string of amino acids that's all bound and wound together. So the neat thing is that this guy can come apart. It can denature or uncoil back into sort of this ribbon form. So it does that, and then each of the subunits gets broken apart. So break apart, break apart, break apart. And so now you just have individual amino acids, so all these dots would be individual amino acids, and that's what gets taken into the blood. Okay, so if you have the blood here, then those get taken into the blood, okay? And then those can travel around and get taken up by cells. So if we have a really large cell here, then we can take a single amino acid and bring it in. So that single amino acid or monosaccharide or fatty acid tail, whatever the monomer is, whatever the repeating subunit is, gets taken into the cell, and then the cell can do something with that. It can go through cellular respiration so that it can get some energy. If it's a protein, cells do really interesting things with those. Because they're not for energy use, they're for lots of other uses. They get rearranged back into a polymer once they're inside the cell. So it's kind of interesting. It comes down as a polymer, gets taken across the gut as a monomer. The monomers get pulled into cells and then reassembled into the polymer. But that polymer that it gets rearranged as won't look like the original polymer. It's going to be a protein that the cell actually needs. So maybe like a transport protein in the membrane, or maybe part of a ribosome in order to make more proteins. So this protein is going to look a lot different than what you ate. Okay, so monomers are the repeating subunits, or the little Lego bricks that are involved in creating these huge polymers. All right, so let's talk specifics. This is a monosaccharide. This is glucose, and so let's write that down. Mono meaning one, saccharide meaning sugar, so it's a single sugar, and this is the monomer for a carbohydrate. The polymer is a complex carb. Not card, but carb. There we go. Okay, so let's go back here real quick. So monosaccharides, polysaccharides, okay? So complex carbohydrate and single carbs. All right, so the purpose of carbohydrate is for immediate energy use. So this is your energy use molecule. And in a cell, if a cell has a choice between a fat or a carbohydrate, it's almost always going to choose carbohydrates. So these are your immediate energy use molecules. Okay, so they're ideal energy for cells. here you can see those sugars all linked together, so you can have really short sugars, 2 sugars, and multiple sugars. So something that you would find in your vegetables and in fruits, something that you would find in candy, baked goods that have added sugar to them, okay, are these guys. And of course, fruits have monosaccharides and disaccharides in them too. So the idea is that we need to be eating more of this. And we'll talk about why a little bit later when we do diet. But ideally, we want longer chain carbohydrates. Short chain carbohydrates taste sweet in the mouth, so you always know when you're eating them. Okay, so this is immediate energy use. So the same concept applies when you take a polysaccharide across the gut. So you eat something like kale, and a polysaccharide comes through on digestion. You break the monomers apart, and the monosaccharide goes ahead and gets into the blood. So real quick, one little heads up about this is that when you eat something that's a longer chain, it takes a while to cut these bonds. So you slowly add sugar to the blood, and you don't get as high as a blood sugar spike from eating things like fruits and vegetables than you would from something like candy or baked goods, cookies, things like that. Because these guys, it doesn't take much. They can just come right across the gut and in some cases be absorbed right in the mouth, and these produce spikes in blood sugar. Okay, just a little preview. Okay, the next thing we want to talk about are proteins. So what proteins are, boy, they basically do almost all the tasks in the body. So your polymer is a protein, and the tasks are numerous. One thing I do want you to know is that proteins are not for energy. So let's write energy, and let's put a big X on it, okay? So I think that most people think that protein in the diet is for energy, and it's absolutely not. The protein is always the last choice. Your cells will only use protein if you are completely out of carbohydrates. And even then, your protein gets converted into a carbohydrate-like molecule before the cells use them. So protein is not for energy. What protein is used for is things like transportation, okay? So moving things in and out of cells. So when we talk about cells in the next lecture, actually, we're going to be talking about transport proteins. So, you know, they move stuff in and out of cells. They're used in protein synthesis itself. So proteins are used to create more protein. Proteins are used in the immune system. Proteins are used for structural support. And in fact, we talked previously in the water lecture about how cells and living things are mostly made up of water, and you can add protein to that list too. So as a human, you are mostly protein and water. So think skin, bone, hair. All of these aspects of you, including many of the aspects of the cell, are protein. So when in doubt, guess protein. Okay, so how do we build proteins? That's from amino acids. And amino acids are the monomer of proteins. And you guys know that. You know that there are essential amino acids, and you can see that all of these are amino acids. So the repeating part of the amino acid is this blue, yellow, red, right? What we care about and what gives variability to long protein chains are these R groups. So these are called R groups, and you're going to see that symbolized when we're in class. So everything in purple is an R group, and R just stands for the reactive group. So you can see here that there's different types of R groups on these various amino acids. That's really what determines the type of amino acid it is, and that's going to be very relevant for us in a moment here. Notice that some of them have charges, and some of them do not have charges or don't have many charges on them. Of course, hydrogen to nitrogen bonds, you know that as a polar covalent bond, but some of these have full-blown charges on them as well, where you're actually able to see these pluses and minuses. Okay. So let's talk about proteins. Proteins are very, very complex, and we will learn about proteins all quarter long. So starting from the beginning, when you go ahead and eat something like protein, let's go back to this slide. Let's go back to our protein that we eat. That gets broken up, right? Cut, cut, cut. Amino acids get taken across the gut and they get brought into cells. So that's where we're beginning our story. These are amino acids that are brought into cells. And what's going to happen is DNA, which is a nucleic acid, and I know we haven't talked much about it yet, DNA that's housed in the nucleus is going to determine the order of amino acids. So if we think about the function of DNA, it's an information coding molecule, right? So it's info storage. And what kind of information is it storing? Proteins. Okay, so the roughly 30,000 genes that are in the human genome are used for creating proteins. So when we talk about proteins, it's impossible to separate them from DNA. So DNA goes ahead and uses these, or it codes for the order of these, and those get lined up in a specific way according to DNA. Okay, so the order here is particularly important. If you end up being off in the order, that's what we call a mutation. That means there was a mutation in the DNA that led to changes in the polypeptide chain, and that can be dangerous. That can lead to disease. Okay, so here they are all lined up. Now let's go back to that concept of an R group. Here the different R groups are symbolized differently, so you can see we don't have any repeating R groups in this sequence. So what this determines is the folding pattern. When you have this particular sequence, with this particular sequence of R groups, what happens is certain R groups are attracted to one another, certain R groups are repelled from one another because of the polarity issue, and that causes it to bend and flex and start to really go ahead and fold. Okay, so this, let's go back here. This is the primary sequence of a primary structure of a protein, and this is called a polypeptide chain. The secondary, and here you can see secondary structure of a protein, we're going to call that alpha helix and beta sheet. So the order of the R groups determines the particular folding patterns, whether it gets spun into a slinky-like arrangement, an alpha helix, or whether it gets bent and folded like an Asian fan. And usually what you have is within the polypeptide chain, you'll have both. You'll have some alpha helices, you'll have multiple beta sheets. These are long, long chains, hundreds of amino acids long. Okay, so we get these folds, and then what happens is you get to this tertiary level where you take all of these twirls and folds and you fold it again. So this is another level of folding. So very, very complex. And we're going to call this globular protein. So the third level is globular protein, and this is where most proteins are functional. Okay, functional just meaning that, well, it can be sent off to do its job. So if we're looking at a membrane transport protein, that's now ready to go get embedded in the membrane so that it can start pumping stuff out or pulling stuff into the cell. Some proteins reach the quaternary structure, something like red blood cells, where you have in a red blood cell 4 of these tertiary structures. So 4 globular proteins that are all connected together around a heme group. So in some cases you reach the quaternary level, but not in all. A lot of proteins stop here. Okay, so let's take a look at what that looks like within a cell. So let's go through what we call the endomembrane system. And the endomembrane system really is a system within eukaryotic cells. And that's because eukaryotic cells are the ones with membrane-bound organelles. So we're going to be talking about the nucleus, which is a membrane-bound. We're going to be talking about the ER, the endoplasmic reticulum. We're going to be talking about the Golgi apparatus. Okay, all of these are membrane-bound organelles. 1, 2, 3. We're also going to be talking about ribosomes, and that is not a membrane-bound organelle. That's a protein. Okay, so those are going to be our 4 locations where we're kind of looking at things. Okay, so let's start in the nucleus. Proteins are coded for by DNA. So inside of DNA, inside these DNA molecules can condense, we'll talk about that a little later in class, into chromosomes. That's how you're used to seeing these. And on those we have genes that are sporadically placed. And those genes code for the order of amino acids. Okay? So one of those genes gets copied. So we need to make a copy of DNA. The reason is because in eukaryotic cells, like I mentioned earlier, that DNA is conserved by this membrane, so this nuclear envelope. Because we have a true nucleus, DNA is conserved. It's just protected from mutations. So we would never send a chromosome or a gene out of the actual nucleus because it would be subject to mutation. So what happens is you make a copy, and some of you guys have heard of this copy, RNA, and then RNA is what leaves. And that RNA is just a copy of the gene for the protein you're wanting to make. So there it is, your RNA, which is a copy of the gene, and this is a disposable copy, and that conserves DNA in the nucleus. And then the next thing that happens is RNA goes ahead and meets up with a ribosome on the endoplasmic reticulum, and this is just huge, massive amounts of membrane here. Okay, so here's the ER, and basically what happens here is you have your ribosome and you have your RNA that gets fed through, and what happens is the polypeptide chain is made from that code. So these different shapes are just representing different amino acids that are strung together. And so this is the primary structure of a protein, and that happens on the ribosome. That's where it's created. That polypeptide chain slips into the ER, and in the ER it actually gets folded. So all the folding happens here. Okay, so secondary and tertiary structure of a protein happens here. So all of your folding. And then the protein, the folded protein, actually buds out. This is a form of bulk transit. And it moves to the Golgi apparatus. And so now we have your protein in here that just came from the ER that's going to go ahead and fuse with the membrane of the Golgi apparatus. And in the Golgi apparatus, a couple things happen. You have short-term storage, and also you have proteins that end up getting flagged for their specific duties. Okay, so that is the endomembrane system. So if you want to see it from a larger view here, in the nucleus, you make a copy of the gene. That goes out to the ribosome, and then the polypeptide chain is made there, gets slipped into the ER, moved, folded in the ER, and then moved to the Golgi apparatus, and then sent out to do its job. So if it was a transport protein, that would just go ahead and be embedded in the actual membrane. Okay, so that concludes our discussion about proteins. Now we're going to look at fats. Okay, so triglycerides are one type of fat, okay, or lipid. So lipid is another word for fat. And you can look at this molecule. It's pretty big. You can see the repeating unit are these actual tails, so from here to here. So that's a repeating unit, 2 and 3. So there's 3 repeating units on this, and of course that's why it's called a triglyceride. And this is the glycerol head in pink here. This is totally nonpolar. This is something like oil or butter. Any type of fat in your diet is going to be one of the variations of this. So in terms of fats in our diet, if we look at the monomer of a triglyceride, which is the fatty acid tail, and we just take one of those tails, we can look at the difference between something that is saturated with a straight chain and something that is unsaturated with a bent chain. Saturated fatty acids tend to come from animal products, and some plant products actually have a large degree of saturated fats as well. What it literally means, though, when you look at the chemistry of the molecule, is that all these carbons are maximally saturated with hydrogens. So H2, 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 all the way down. If you've had chemistry before, you know that carbon can form 4 bonds, and only 4. So there's 1 bond, 2 bonds, 3 bonds, 4 bonds. So it's maximally saturated with hydrogens. That is not the case for unsaturated fatty acids. So here you can see a double bond and only 1 hydrogen for each of these carbons. What that double bond does is create a kink in the chain. And you know, at room temperature, you have oil being liquid and butter being solid, even though they're the exact same polymer. So that's kind of odd. The reason is because of molecular motion. These guys are, you know, they can be packed together. So you have chains, you have multiple fatty acids, right, that are together here, and they're side by side by side by side in butter, and they're crammed in there, and there isn't much room between the tails. So, you know, temperature is a measure of molecular motion, and there's not much molecular motion because they're packed in there. That's different here. So there's actually room between the tails, and so there's movement, and you're liquid at room temperature. So we'll talk later about how this relates to diet, but this is just a foundation for you to understand what is saturated versus unsaturated. We will talk about hydrogenated oils. These are partially hydrogenated oils, and I'm going to save that for later. Okay, the second type of lipid, or fat, is a phospholipid. And phospholipids are involved in cell membranes. Okay, so that's their main role. And they're kind of unusual because they have a head on them that actually has charges on it. So see that plus, see that minus? That means the head is polar, or soluble, or hydrophilic. And the tails are just like tails on the fats you eat, right? CH2 all the way down. Oh, here's a double bond, so this is actually an unsaturated phospholipid. You can have saturated and unsaturated and phospholipids too. And the tails are nonpolar. So that's kind of unusual. So what happens when you put phospholipids in water, let's say in a cell, is that they orient themselves in this way, where the tails like each other in here, and the heads stick out in water, because these don't like water, so they group towards one another, and the heads stick out in water because they like water, because they're polar. And this is fantastic, because if you keep this going around, that would be a cell membrane for an entire cell, and that means that only some things can get in and out freely, because now you have a hydrophobic domain. And if most of what a cell has are things like ions with positive charges on them, then they're going to do fine with the heads, but they're going to get bumped out by the tails, and they're not going to be able to get in. This gives the cell regulatory control. So let's look at a cell membrane. A cell membrane is not just phospholipids. There are actually quite a few proteins embedded in the phospholipids that are involved in transport. So the phospholipids themselves aren't involved in transit. It's the proteins that are embedded in the phospholipids that are. The phospholipids just keep things out or in. There's also an extracellular matrix that we're going to be talking about for animal cells, and yeah, we'll talk about that with atherosclerosis. Okay, last one is nucleic acids. So this is the polymer, nucleic acid, and this is the monomer, nucleotides. So if you look at a nucleotide, it involves a phosphate group with negative charges on it. So there's charges here. It involves a sugar, which has O to H bonds, and it involves one of these nitrogenous bases. So the sugar and the phosphate remain constant, and it's the base that changes. Okay, so this would be like our R group on the amino acids. So what this tells us, we have a charge here, and we have O to H groups here, and we have N to H groups in all of these, and that means that DNA likes water. So that's a good thing. DNA likes water, and it's housed in the nucleus, and it has no problem being in an aqueous solution. So DNA, its function is to code for protein. So it's an information storage molecule in its function. So I talked earlier about chromosome and genes. Genes are coding for proteins. So a gene sequence is a sequence of nucleotides that are coding for a protein. And that is what a gene is. Okay, so a sequence of nucleotides that are coding for a protein. So these nucleotides, since the sugar and phosphate remain stable, we're not really concerned about that. What we care about is the base. So what we want to know is, what is the order of the bases? So what is it? Is it A, T, T, G, C? Because that could code for one protein. Or is it C, C, G, A, A? That could code for another protein. So you have variability here. In mixing up these bases, you can code for different proteins, and that creates differences, right? That creates different proteins, and we have millions of proteins that the human body can create, and that's dependent upon the order of the bases. So that's actually where the information is in the sequence of the bases. One last thing about nucleotides. So there's one exception here. There's a nucleotide called ATP, and it does not form nucleic acids. It stays as a nucleotide. So here's your sugar, there's your phosphate, and here's your nitrogenous base. So adenine is the nitrogenous base that's used. And these phosphate groups, you can see there's more than one, which is different from this slide where I was just showing one. That's okay. It's still considered a nucleotide, and we're going to talk about the importance of these phosphate groups, but this is basically cellular energy. And it's called and it's a single nucleotide. Okay, that concludes the lecture on organic polymers.